Okay, this is lecture 3 in Emergent States of Matter. Last time we talked about emergent patterns far from equilibrium, and we talked about the question of what happens when you have a phase separation, two phases that are immiscible. They don't want to be near each other, so orange and blue in these pictures here. And as a function of time, the system gradually coarsens like this as you quench the system and then wait. The other thing that we talked about was if I took the initial condition and then just expanded it, we could see that the sequence of pictures you get from expanding also look just like what you see with the time evolution. And so you get the idea that the time evolution is somehow scale invariant in a way that we want to quantify. And in a minute, I'm going to extend this to the topic of this course, which is systems with continuous symmetries. So these pictures here are where you have a boundary between orange and black. So this would be true if you had, say, a system with a discrete symmetry, like up-down symmetry in an Ising model. So we're going to see in a minute what happens when you have a tensor or a vector order parameter. So what you measure is the X-ray or neutron scattering function from a material. So in the data that I'm showing you here, these are X-ray neutron scattering from a copper manganese alloy. So it's doing exactly the process that I showed you in the previous slide. And so what you're measuring is the structure function as a function of wave number q. And we'll talk about what the structure function is. It's basically a 2-point correlation function. It just tells you how much scattering there is at a particular wave vector. I'm not going to go into this in great detail, but the thing I want to show you is that these are graphs, S, the scattering function, as the Fourier transform of the correlation function, as a function of wave number for different times. And you can see that there is a peak in the structure function. And that peak in the structure function corresponds to a wave number. And because it corresponds to a wave number, it corresponds to a length scale. So what length scale could that be? Well, it's exactly the length scale that you see here, which is the characteristic size of the domains that you see. So we can quantify this picture by doing neutron scattering and looking at the correlation function. Now, what I've shown you on the right-hand side is the experimental data. And what we're doing is we're taking the wave number at which the structure function or scattering function has a peak. We'll call that Q max. And you can see that that peak goes to smaller and smaller values of wave number as the time increases. And that corresponds to the fact that the domains are getting bigger. And so the wave number, which is the inversely proportional to the size, is getting smaller and smaller. So then what happens is if I take the structure function, multiply it by the time-dependent wave number, the peak wave number value raised to the third power, and I plot the horizontal axis as being wave number over this peak value, so I've scaled it in that way, then all the data for all these different times shown here and shown in these earlier time graphs, all these data collapse onto one universal curve, and that's the one shown in the top picture here. So this data collapse is really quite fantastic. It's the non-equilibrium analog of what we saw happening near a critical point for a magnet. And so, and this behavior would in principle be universal and could be computed. However, in practice, you have long-range effects that can make this non-universal, for example, lattice strain effects and so on, which are very important in real-life solid-state alloys. So, this is the how one puts a picture on this idea that I get scale invariance in time as opposed to scale invariance in space, which you normally see in equilibrium statistical mechanics. Now, one can model this in a very simple way, and I'm not going to go through this in detail. The way that material scientists did this in the past was writing down a differential equation called the Cahn-Hilliard equation, and then just solving it on a big computer. But you can do much better than that by just noticing that really the potential energy of the system, the free energy, has two minima corresponding to the two coexisting phases. And so the idea is this: if I take a system and I coarse grain it, I look at it on a large enough length scale, then I can write down a dynamical variable that tells you something about, say, the concentration of one of the alloys in a particular region of space of order the correlation length in size. So the idea is this: there's really three different fixed points of the dynamics. There's one fixed point that corresponds to the one of the coexisting concentrations of the phases, the oil-rich phase, let's say in a salad dressing. There is the vinegar-rich phase, let's call that beta, and then there's the phase where they're all well mixed, which is homogeneous, and at high temperatures is the stable state. But at low temperatures, the system undergoes phase separation, and so the well-mixed state, where there's no separation into two phases, becomes unstable, and the system goes either into one of these potential wells or the other. And so what the idea is that we do, and we started doing here at Illinois maybe 30 years ago with a work that Yoshi Ono and his student did, Sanjay Puri, was we started thinking of these things as renormalization group flows, and just writing down model dynamics that couples regions in space with maps that have these fixed point structures, and then imposing conservation laws. So I'm not going to go through this in full detail, and I don't expect you to understand the details of this. And later on in the course, we can talk about this some more. Maybe some of you may want to even write a term paper about it. But the point about this is that we have a pattern that emerges in a universal way. And in fact, it turns out that the way in which the domains grow, it has a power law scaling in time, and that power law is universal, just like the power laws you get in critical phenomena. And we use that to develop very fast numerical methods that do not do the usual thing that we talked about last time, where you take a continuum, then you make a grid, and then you look at the convergence as the grid spacing goes to zero. That's the normal thing you do in numerical analysis. What we're doing here is we're taking advantage of universality and the normalization group concepts, keep the spacing in time and space as order one, in other words, not going to zero, and devising maps that have the right fixed point structure, and hoping that if you want to capture the universal behavior, you don't need to capture the excessive realism of the actual process. So I'm not going to go through how we construct these maps in detail. There's lots of tricks here and interesting technical points that I can talk about some other time if you wish. But the point about it is that it produces the results that you saw over here, and it completely reproduces the scaling that you see in experiment. And let me show you another example of that. So basically the summary point I want you to learn from this, without going into detail, is that if you make a numerical algorithm that models the same thing as a differential equation, both should be in the universality class as the physical process, and so you would expect that it would give you the same results. So so this is a completely alternative picture to the way that numerical analysis is usually done. So let me give you an example here. This is what happens now when you have an order parameter, which is a tensor, and that's sort of one of the things that we are going to be focusing on in this course, which is systems with continuous symmetries described by order parameters that are not scalars, like plus or minus, like you get in, say, an Ising model, but order parameters that are complex numbers, that are vectors, that are tensors, and they have their own order parameter structure. And in the very first homework set of the course, you're going to be looking at what kind of interesting features can arise from the interplay between the space-time symmetries of a system and the internal symmetry group of the system. So here's a very simple example of that. This is liquid crystals. Liquid crystals, as you know, are just rod-shaped objects that float around in space. They're called liquid and crystalline because their centers of mass may be random, like a liquid, but their directions that they can point in are ordered, and so they're halfway between liquid and crystal. And liquid crystalline systems, as you'll quickly figure out when we get into the specifics, have topological defects. And so here what we're looking at is a liquid crystal system through cross polarizers, and we're looking at the light that is transmitted. Basically what happens is when light goes through such a system, its direction of polarization gets rotated. And then if you polarize the light with a plate below the system, and you polarize the plate above it in the 90 degrees direction, then what you see transmitted is the light that has been rotated by the orientational order parameter of the system. And you'll get to study this in a bit more detail in homework. So these kind of bow tie structures here, these things that you can see at the edges here, these things are monopoles. These are topological defects, kind of like the monopoles that you've heard about probably in grand unified field theories that describe the matter in the early universe. And what you can see happening is that instead of having domains that grow in size, you can see that at the beginning you have lots of topological defects, these monopoles, identified here by these bow tie-like patterns. And as time proceeds, you get fewer and fewer of them. So they get further and further apart. And what's actually happening is that nearby topological defects are running into each other and annihilating. And then the only ones that are left are the ones that are not annihilated. And so it looks like the average spacing between these topological defects is increasing. So this is what you see in a microscope, a type of Schlieren photography or phase contrast microscopy. But in fact, these pictures here are not experimental pictures, although they look exactly like experimental pictures. They're actually computer simulations that we did of this system using a tensor order parameter. Now here is experimental data. And this was an experiment that was done in 1995, a few years after we made an experimental prediction, which I'll show you in the next slide. So what I want you to see at the moment is just this. We have liquid crystals that are undergoing a phase transition. And as they go through the phase transition, the coarsening is reflected in the mean separation between the defects. So here there's too many to really see. By the time you get to 32 seconds, you can see there's many fewer of these topological defects, and the distance between them has grown, just like you saw on the previous picture. So now the question is, does this system have that same kind of scale invariance and time that I showed you for the magnetic data? So what we did was, in theory, and this was done in 1992, before the experiment was done, we simulated this type of experiment, and we discovered that you should get the data collapse of the correlation functions as a function of distance. So this is the scaled correlation function on the vertical axis. This is scaled distance here, and these are data taken at a variety of different times. Just like the neutron scattering data on the copper manganese alloy was looking at the correlation function as a function of wave number at different times, and when scaled in the right way, all the data collapsed onto one universal curve. So what you see here in the black squares is what we got, the universal curve we got from our simulations. The dots and squares and triangles and crosses and plus signs, those are the experimental data from these pictures digitized and plotted in the way that the theory predicts. And you can see that they fit within scatter very well the predictions of the computer simulation shown in black. And then the solid line is an analytical theory that was done by one of my former students and Gene Mazenko when he was a postdoc. And you can see that without any adjustable parameters, the experiment agrees with the simulation and very closely, except at very small distances, with the analytical theory. And so this again is a remarkable thing. You've made a non-trivial prediction of an extremely complicated space-time pattern process using a computer program that in fact is 5 lines of code. and and again, and you know, here is essentially the pseudocode for the computer program. The only difference is that in the code that we ran, the order parameter was a complex number rather than a scalar. So what we've learned is that making a discrete model on a lattice without doing anything to do with grid convergence or all the things that you would learn about in a numerical analysis course, at least a conventional one, you find that you still, with a minimal model, correctly capture all of the features of the data collapse, with scaling laws, no adjustable parameters. And the only thing that it doesn't do for you, as we talked about last time, is that it doesn't tell you, if you want to know exactly how far apart two topological defects are after 13.4 seconds, and you want to know the distance in microns, we can't tell you that. We can only tell you about the scaling laws, but if you tell me what the spacing is at a particular time, then thereafter one has that one initial condition and one can then compute anything you want. So what I've shown you then is that when we make predictions in physics, they rely, the reason that they work in physics is that they rely on the existence of minimal models, and the predictions that we make are typically parameter independent and universal quantities. And a lot of the ways where physics has been successful is because the predictions don't depend sensitively on details, and so one can avoid excessive realism to be able to make minimal models that capture the important quantities. Okay, so that's how we use emergence and how we use these ideas of minimal models to do something in practice. Now I'm going to skip through this. I'm not going to show this. If we have time at the end, I will talk about this. That is my current ongoing work. I want to talk about something different now, which is about biological systems. So before I do that, are there any questions about the minimal models and the idea of emergent pattern formation?
Hey, Professor. I have a question so you said that scale invariance in time evolution, that you see scale invariance in time evolution. But where in the Hamiltonian do we add scale, like a scale parameter?
You don't. It emerges.
So the entire Hamilton. So
remember that when we're looking at a dynamical system, we're not really doing equilibrium statistical mechanics. So you may start off with a Hamiltonian, say describing spins that just point in a plane, or liquid crystals described by the tensor order parameter. And then you quench the system, and then you evolve it forward in time using, say, Monte Carlo simulation or some other method like the method I showed you. And the length scales that you see emerging, the pattern that you see emerging, and the scaling of those length scales with time is not put in at all. It's an emergent phenomenon.
Oh, okay. But because what I was thinking was that since you said that we saw as an emergent process, we saw that scale invariance was seen in time evolution. So I was thinking that, hey, in the Hamiltonian, there was no such scale parameter in any, I mean, I don't know if I remember any Hamiltonian with like explicitly scale put in. So we always knew this fact that there's scale.
Yeah, the system self-organizes and creates the scale itself. You can't know that by looking at the Hamiltonian. In fact, it's a property of the dynamics, not a property of the Hamiltonian. The Hamiltonian doesn't, on its own, specify the dynamics. So, for example, in the system that I was showing you, what we were solving was the time-dependent Ginzburg-Landau equation, which we'll talk about in the class. If, on the other hand, I had an order parameter that was conserved, then I would be using a different equation and an equation of motion that reflects the conservation law. And so one has different universality classes for non-equilibrium dynamics based on symmetry groups, based on conservation laws or non-conservation laws. They're somewhat, it's a different classification than what you have in, say, critical phenomena and equilibrium phase conditions.
Thank you, Professor.
Okay. All right. So I want to talk a little bit about biological systems and about levels of description in a different way. So let's talk about this statement. Theoretical physics is so successful because, okay, why is theoretical physics so successful? So anybody want to make some suggestions?
It's able to make like predictions and align whatever model with experimental data.
Yes, that's right. So what you're saying, who's that? That was Adam, was it talking? Yeah. So what you're saying is that we have a method in physics whereby we have this sort of virtuous cycle, which is an interplay between experiment and theory. Theory making predictions, experiment testing them, and then refining the theory, and then repeating that whole process many times, and so getting a good result. That's definitely true, and I'll probably say something more about that in just a minute. So yes, so the interplay with experiment is critical. Completely agree with you. Anybody else got anything else they want to add?
Well, since you're dealing with minimal models, you don't really have to add so many details of specific systems, so you can apply it to a bunch of systems as long as it fits that minimal model.
Exactly right. Yeah, that's. I'm trying to see who answered that. Was that Maisha?
Yes, that was Maisha.
Maisha, thank you. Yes, yeah, exactly right. So we have a perspective of trying to model things where we think the way we model will work. We haven't been very good at doing that, say, for economics, for example, where we don't know if this minimal model picture would actually work. It's not necessary that it should work. It only works in simple cases. So one of the things I would say is that we only ask simple questions, and that's a way of saying we ask simple questions which can be answered, or we hope can be answered by minimal models. Another thing I would say is that we ask questions at the top level of description. In other words, we only, if necessary, venture down into lower levels of description where one needs excessive realism. So it's really just another way of saying what you were saying, and that key point is that they don't depend on the details at lower levels of description. So we ask simple questions in this particular way. So not every subject has the ability to do that. Not every thing that you want to know the answer to is it mandatory that can work. So let me give you an example here, and I'm going to put in a kind of joke, really to exemplify the points that both Adam and Maisha said in their answers. So I want to compare and contrast in a rather joking way, but also makes a serious point way, Newton's laws versus what's known in evolutionary biology as purifying selection. So let's talk about the nature of explanations. So here is an apple tree, and this is actually Mr. Newton's house. And Mr. Newton is saying, so why did the apple fall down? And we all know this is a famous story of Isaac Newton coming up with the law of gravitation and so on, seeing an apple fall down. Whether it did or not, I have no idea. But anyway, that's the story. So there's several different explanations that one could imagine. So the physics answer is this. Well, let's make a guess for what the law of force is between two bodies. So for various reasons, Newton guessed his universal law of gravitation. You write this down, you combine it with F equals ma, you do some calculus, and you predict that the result should be an ellipse, and that applies to the apple, applies to the moon, the planets, the comets. You can make many predictions, and that works very well. So we're lucky that we're able to do it in this way. Here is a joke. but it's a parody of an answer that you sometimes get in evolutionary biology. So here is a joke answer, which is, well, there's two types of trees. There's ones where the apples fall up, and there's ones where the apples fall down. So if you think about the type 1 tree, well, their apples will fall up, and so they will go off into space, and they'll leave no seeds in the soil. And so those trees will die out. And that means that the only ones that you will see are the trees where the apples fall down. And so that's why apples fall down. Okay, so that's a kind of evolutionary biology answer. And if you mean this question is so simple that we kind of know what the right answer is, or the most useful answer. They're both guesses. The physics answer, in some sense, is better because it ignores certain details. It uses mathematics to work out the consequences. It can be systematically improved. It's quantitative and not binary, and it makes predictions that can be tested quantitatively. So those are all the things that you guys mentioned. And so, but the real point is that if you really want to understand about biology, it's incredibly complicated, and it's not that biologists aren't smart enough to answer those questions. Physicists could. It's absolutely wrong. We are only successful because we ask mind-numbingly simple questions. So let me give you an example here, and again another sort of joke thought experiment, just to try to ram the point home. So here's some questions that you might ask. Can you fix my car? Who would you ask to do that? And what would happen if you tried to ask a physicist? Well, so, your car's broken down. How do I fix it? Well, you'd ask a mechanic. Would you ask a physicist? Definitely not. That would be asking for trouble. If you ask the question, how does my car work? Well, the specialist might, not really be able to tell you in the detail that you would want. A physicist might mumble something about the Carnot cycles or second law of thermodynamics or something. Probably not completely accurate, but at least something about that. You might say, okay, well, my television's broken. How do I get that fixed? So again, you call an electrician or something like that. Would you ask a physicist to fix it? Well, if you have very good insurance, maybe you would, but I wouldn't recommend it. If you ask a physicist, how does my TV work? The electrician might not be able to really give you a detailed answer. I know as a fact, because when I was a kid, this exactly happened to me, and I did try asking an electrician who came to our house what electricity was and how things worked. If you ask a physicist, they'll mumble something about Maxwell's equations. So, okay, that's true. If you say, well, how do I fix my sick living system? I'm feeling ill. You'd go and definitely ask the doctor. Would you ask a physicist? Well, definitely not. Although I've got a qualified to that, which I'll show you in a minute. If you ask, how does my living system work? The specialist will say, I can tell you about the parts and how they fit together, but really, do we understand it? At some level we do, at some level we don't. Does a physicist know the answer to that? Well, at some level we know a little bit, some level we don't. We know a few more tools. So this is just to show you that there's really some questions that really you can't really address as a physicist. I just put this slide in to make the point that you might ask a physicist about living systems out of desperation. In this particular case, this is the National Cancer Institute. A few years ago, they started a program called Physical Sciences Oncology Initiative, trying to basically say, okay, well, we give up. We don't understand cancer. Our last resort. We'll see if physicists can be involved and do anything useful. Whether they can or not, the jury is still out. And say, putting it politely, this was a very interesting initiative, and a lot of good things did come out of it. And I was involved in helping some good people do some interesting things in this area. So I mean, there's no question that there's a merging of disciplines, and there's a lot to be learned from that. But these are very, very difficult questions. So we don't have an explanation for how living systems work, or even why they exist. And this is a fundamental question in emergent states of matter. And I don't know whether we'll have time to talk about it in the class, but some of you may want to talk about it and in your term essays. And I'll try to say something about it near the end of the semester, but I want to say a few things about it right now. So the real question from my perspective as a physicist interested in biology is, what would an explanation look like? You know, how can you make this question sharper so you can really try to use physics tools to understand it? And I'd say it's in a different way, which is when we answer questions in physics, we answer them at a top level of description. We look at the symmetries, we look at the order parameters, we try not to get into excessive realism. So the question I want to ask is this: when we think about biology, we think about biology as being molecular biology, biochemistry, lots of polymer molecules and things like this. It's a very molecular type of understanding. And indeed, a lot of the big advances in biology came in the 1950s and 1960s through the advent of molecular biology, in which physicists played an important role. So my question is this: if we try to have a top level understanding of biology, if we had such a thing, how useful would it be? And I want to argue that it actually could be useful. So let's go on and talk about what I would like to call provocatively as universal biology. So let me again try to motivate this with a joke. So here's a headline that you may have seen 5 or 6 years ago. If you are following NASA, you would see, for example, a headline like this: "The mystery of the missing waves on Titan." So Titan is a moon in the far solar system, and it has lakes and not water, but it has lakes. And now the question you want to ask is, well, there's an atmosphere, there's lakes, shouldn't there be waves on that? And so where are they? And so there was the big mystery. And just in case you lost sleep over it, don't worry, because about 6 months after this headline came out, here was the final answer. Cassini, a wonderful satellite mission, which is my favorite satellite of all time, Cassini spies the wind-rippled waves on Titan. So you can sleep at night, mystery solved. Okay, here's a headline that you didn't see: "Mystery of the missing life on Titan." And you certainly didn't see this one: "Cassini spies the long-sought life on Titan." Okay, so why is one of these headlines a real thing and one of them a joke? Well, actually, this isn't quite a joke, because this mystery of the missing life on Titan, Cassini spies the long-sought life on Titan. Actually, if you replace Titan by Enceladus, a moon of Saturn, actually, it's not impossible that it has spied life on Titan. So let me just tell you very briefly about that. The biggest advance in our understanding of the solar system, I would say, in the last 5 to 10 years, has been the discovery that the outer worlds of the solar system are covered in ice, but beneath the ice are oceans of liquid water, global oceans of liquid water that covers the entire planet underneath the ice. And we made those discoveries from the satellite Cassini, which has been able to look at the wobble of the satellites as they move around Saturn, and from that one can detect what they're made of, in just the same way that you can take a hard-boiled egg or a soft-boiled egg and tell which is which by looking at the way they rotate when you spin them round and stop them. So one of the great achievements that Cassini did was this: Enceladus is a small moon of Saturn. It has a global ocean of liquid water, kept liquid by tidal forces as it rotates around Saturn. And that water actually can get to the surface. And how does it get to the surface? Well, there's a huge ice cap around the moon, but there's cracks in it. And out of one of those cracks comes a fountain of water from the inside of Enceladus. And that water, you can send a spaceship, you can send Cassini through it, and you can sample the water and look at the chemistry of it. And in fact, we see that the water chemistry looks very much like the water chemistry that one sees at a hydrothermal vent underneath the ocean. In fact, in the alkaline hydrothermal vents, where many people believe that life on Earth may have started. And so actually, it's not a complete joke. We might actually have been able to see some of the evidence that there is non-equilibrium behavior of the water under the ice caps of Enceladus, indicative of non-equilibrium processes that describe early life and microbial life. So it's very exciting. And if, you know, this is a field, astrobiology, which is really becoming a quantitative science and is really a tremendously exciting one, which I spent some time on. Okay, so that's the idea that, you know, what do we know about universal biology? Well, pretty much everything we know about it is on this slide, so basically nothing. So to try to tell you how the ideas in this course could be relevant to biology, I'm going to talk not about universal biology, but about universal computation. Okay, so what do we mean by universal computation? So that's a computer, right? Pretty much like the computer that we're all using right now to listen to this lecture or to give this lecture. Here's another computer. This one is made out of cogwheels and springs and levers, and was built, partially built by Charles Babbage in the 19th century. And you can see it actually operating in the Museum of Computation in Mountain View in California, and also at the Science Museum in London, where it's been restored. So this is a machine that, if it had actually been built, would have been a universal computer. It was programmed in a completely different way, but in all essential purposes, it was the same thing as a modern day laptop computer, just implemented with cogwheels and springs. So the point is that a computer is not a shiny chunk of glass, plastic, and silicon, nor a bunch of cogwheels, springs, and levers. It's an abstract concept. What makes these two things the same is that any computation that you want can be performed by them. This, to perform, you know, complicated calculations, you can easily do them on a modern day laptop. To do them on a machine like this would have required a huge device that would fill, you know, the Albert Hall or something like this, and would take, you know, millions of times longer, but it would still be able to do that. And the reason that we know that this thing is a universal Turing machine, meaning you can prove using the theory of computation that is capable of being programmed in any way, is that it has the ability to have loops and conditional branching. And so that means that one can prove that this is indeed what's known as a universal Turing machine with a von Neumann architecture. And so the point that I'm trying to make then is what's universal about computation is not the machine that it's built on. Whether it's Windows or Mac is not the point. The question is whether it is implemented with transistors, cogwheels and springs, or even thermionic valves. What's important is what it actually does, which is in terms of the software that it executes. So the medium is not the message. This guy here is John von Neumann, one of the great mathematicians of the 20th century. Many things that he did, even at the birth of quantum mechanics. This is him in the 1950s at the Institute for Advanced Study at Princeton, where he decided to take this abstract mathematical concept of a Turing machine and ask, how could you actually build a computer out of parts that are unreliable? So remember last lecture, we talked about how do you make a solid out of polymers where the polymer chains are floppy and they're not rigid themselves, but you can make a solid out of them. And we talked a little bit about polymer solutions and polymer gels. Here you have the question of, I'm making a computer that has to work, has to be able to give you an answer and do a computation, but the individual components of that machine are subject to failure. And he worked out the mathematical theory of that, and then got a bunch of engineers, and they worked at the Institute for Advanced Study to actually build some of the world's first computers. And they were doing this in order to do weapons computations, but other people use them for something a lot more interesting. So what I've what I've just told you is that when we think about biological systems, and we talk about things as complicated as a computer or a living system, what we really mean when we say that we understand it is that we understand why the thing exists in the first place, and then we understand its specific instantiation. So let's go back to a simpler concept that we've had in the course. I showed you about the universality of the behavior of magnets near a critical point. And what we saw is the following. We could understand physically why you get magnets, why you have a critical point, why you have a phase transition. And we can also understand, at least maybe you and I can't, but somebody who's a materials scientist would be able to understand, if I had a particular material, they would be able to calculate from electronic structure or something like this, exactly how that particular realization actually works. So I'm just distinguishing the sense in which we understand physical phenomena by the fact that we understand why they exist in the first place, and then we can understand specific instantiations or specific realizations of them. So in computers, we understand why a computer can exist in the first place. That's a Turing machine capable of Turing computation. And then we can understand how to make specific ones. If you're an engineer at Apple or at Lenovo or something like this, you can design the specific machines that do that. Now, when we're talking about living systems, what is the sort of essential feature of a living system that defines what it does? And I'm going to argue that the real question, the real kind of new law of physics that emerges when we're looking at living systems is that matter can, for reasons that we don't really understand, self-organize hierarchically to create replicating and evolvable structures. So in other words, everything that we have on Earth is made of dead stuff. But somehow life, those atoms organized, and they organized in a hierarchical, somewhat scale, not in a scale invariant way, but certainly different levels of description. And they created living systems which are replicating, they use energy, and in particular, they evolve. So the interesting feature about life is not that it replicates, not that it uses up energy, but the fact that it evolves in an open-ended way, complex systems. So I would argue that the essential feature is that these are dynamical systems that can reprogram themselves. So the idea here is this. If I have a dynamical system that we have in physics, you tell me what the Hamiltonian is, you write down the equations of motion, and everything is done, and that's the end of it. But when you have living systems, they're capable of rewriting their own computer program. So it's basically saying that the program is the data and the data is the program. So let's just unpack that a little bit. So this guy here is Niels Baricelli. So he was a scientist, I think he was a physicist, at the Institute for Advanced Study when von Neumann was building his machines. And after they were done, during the day doing bomb calculations, he would go in and he had this idea that I could make computer viruses in this computer, things that would replicate, things that would have errors, things that could then have evolution. And so he ran numerical experiments on these very simple prototype computers, showing you something here. It's inscrutable to know what this means, but what this is one of the outputs of one of the simple first computer virus experiments that Niels Baricelli did, looking at computer viruses and how you can make ecosystems of them and how they can co-evolve. And so the idea was that these computer viruses had their own code, which was the stored as bits in the registers of the computer, and their action was to rewrite their own code. And sometimes it produced complete garbage and they died, and sometimes it produced things that were good and then could replicate. And so this process happened in the computer. It's exactly the same process that happens with viral evolution in real life. So basically the idea then is that evolving living systems the living systems have this capacity to evolve, and that is the defining feature of them. And so what that means is that they are able to reprogram themselves, and by reprogramming themselves, they can create new functionality, and that new functionality might enable them to adapt better to whatever external environment they are in, and so on. And so this is the idea that you can have systems that rewrite their own code, and so they are self-programming, evolving systems. Okay, so here's something which is very speculative, and I can't really tell you that this is something that is true, or even that I know how to prove it if it is true. But the idea is that such systems that reprogram themselves, their future evolution is unpredictable, perhaps even Gödel under Seidel. And by that, what I mean is that you can't, in a physical system, you can tell where it is going to evolve to, whether it is going to pass near any particular point in space or not, sorry, any point in its phase space or not. That's what really is one of the foundations of statistical mechanics and equilibrium. But if you have a dynamic system that can rewrite its own program, as it were, you don't even know what degrees of freedom will represent it in the future. And that means that such systems are very difficult to control. Okay, so why do I talk about this? Because in fact, this is really one of the hardest problems in biology, which is how do you control living systems? So let me, before we get into that, which, and I do want to end this lecture with talking about that point, let's talk about universal biology. I've talked about universal computation, so let's talk about what the problem is in universal biology. So we are actually given, as biological scientists, those of us who are, we are given the inverse problem, not the usual problem. Suppose I said to you, here's a computer, so here's my laptop or my cell phone or something like that. Could you work backwards from that? If you are, an alien being from another planet, came to Earth, I gave you this thing, could you work backwards from looking at that, taking it apart? Could you work back from that and deduce Turing's abstract theory of computation? And I think you'd find it pretty hard to do. I think it'd be, you might be able to figure out what the thing does, but if you didn't know anything about it, it would be pretty hard. So in biology, we're given that same problem. We look at living systems that we have access to now, and what the physics question is that would really underlie this is what mathematical abstraction are living systems an instantiation of? So let me just recap and unpack that question. I told you that every computer that's ever been built is an instantiation of an abstract theory due to the mathematician Alan Turing and von Neumann and others who developed the theory of computation back in the 1930s. So we understand in principle how one can have universal computers. Any kind of thing has an abstract theory behind it. So the question we want to ask is, what is the analog of the universal theory of computation for living systems? And how can you answer that question? Well, all we know is what we see in living systems around us that we have access to experimentally. And so what we'd like to be able to do is say, can you do the inverse problem? Can one figure out what that abstract theory would look like based on what we have access to, which are only instantiations of it? It would be like saying, if I give you a whole bunch of materials from some solid state physics lab, and I just look at them, and some of them may be, yttrium, barium, copper oxide. Some of them might be, some other alloy. Some of them might be pure crystals. And I say, can you work backwards from that and work out the theory of, say, quantum mechanics or the band structure of electrons or something like this? Very hard to do, just given instantiations. But that's what one would like if one wanted to have a top high-level description of living biological systems. Okay, so that's very philosophical. So now I want to show you that it's not pure BS. So I want to show you now that there are universal things in biology, even though everybody knows that everything in biology has an exception. And so what we think about as the characteristics of biology, there are universal things, there are simple things that one could imagine asking questions about and trying to understand. So let me give you an example of a successful abstraction in biology. And my favorite example of that is the universal genetic code. Okay, so I'm not going to go through this in detail because there's lots I can tell you about this. So I'm going to skip through some of the things I want to say about it. The universal genetic code is literally a code. It says, if I know this letter in the first column, this letter in the second column, and this letter in the third row, I can tell you what those three letters will code for in terms of amino acids that make up proteins. So if I had U, and U, that would be the code in your genome for the amino acid phenylalanine. And if a ribosome translating your DNA or mRNA into proteins encountered that string in a codon, it would produce that amino acid. That's how you're made. And this is a code that is universal. Every living system that we know of has exactly this code with very small variations. One of the things that was discovered about 20 years or so ago is that this code is optimal in minimizing errors in some sense. In other words, if you have a mistake and you get the wrong amino acid, then the wrong amino acid you get will be, in most cases, good enough to do the task of the amino acid that you should have had. And so there is a hidden structure in this code. And the other thing that we learned from doing computer simulation is that this particular code, I'm not going to go through this in detail, evolved to have the property of uniqueness and optimality. And it did it through a mechanism that involves energy, information flow between living systems through what is called horizontal gene transfer. Gene transfer between organisms that are not related, not given to your descendants, but between contemporary living organisms. So it's the mechanism by which antibiotic resistance is being spread amongst the bacteria that are in your stomach right now. And it is a very powerful force for evolution. So I don't want to go into my own work on this. I want to talk about some other things, but this has enabled us to understand, in fact, from dynamical systems theory and understanding this, how it is that we have a universal genetic code underlying all life on Earth, and I think life elsewhere, if one were to find such a thing. So that's one example of universality. So here's another one. I'm going to show you some other data here. This is the statistics of gene expression. So if you have cells that are genetically identical, you might think that they all produce exactly the same proteins, but that's not true. They produce different numbers of proteins, and so we say that gene expression, the production of proteins, is a stochastic process. And in fact, it's a very interesting statistical mechanical process, because when you have statistical mechanics, say in the course that I teach in equilibrium, we're usually dealing with Avogadro's number of particles, a huge number, 10 to the 23. But in a cell, you might have hundreds or thousands or millions of particular molecules that you're interested in. And so you're not in the thermodynamic limit. So you have lots and lots of fluctuations. And people like me are interested in trying to understand the fluctuations you get inside living cells. So you might ask, well, what determines how much of what a protein is in a cell, and can you measure it? And the answer is you can measure it, and I'm not going to go through. And you can measure how many copies of a particular protein you will find in particular cells. And what you find is that when you do these measurements, you find all the data collapse onto one universal curve, showing you a sort of universal probability distribution that tells you that controls the stochastic process of producing proteins from genes inside cells. So this is something clearly universal, and we have some idea about where this comes from. It comes from negative feedback control between the growth of cells and the way that the reaction networks interact inside a cell. Okay, so this is an advanced topic. I'm not going to have time to go into it, but just by observing these universal scalings in cell biology, one can then learn something about the large-scale way of describing such a system. Here's another example that I like, and this is you take a methanogenic bioreactor fed with glucose. So basically, what that is just go into your garden, pick up some soil, stick it into a plastic box, add water, add some glucose, and then look at how much methane comes out at the end. And what you find is, if you do this experiment in a controlled way, is you find that the amount of methane that is produced is basically a constant in time, and you can run such a reactor for a year or 2 years or 3 years, and it will always produce the same amount of methane. And so you might think, I've got just a bunch of bacteria in here, different species of bacteria are just in equilibrium with each other, and they're just sitting there metabolizing the glucose that you are feeding them. And that would be a perfectly normal assumption. Nobody would disagree if you assumed that. The thing is, it's completely wrong. And what Fernandez et al. Discovered by accident, and nearly didn't even publish this, was that when they studied, they couldn't really do genomics properly in those days. They did something like it. What they were able to find, and these different colors here represent different types of bacteria, what they found was that the populations were continually fluctuating. They found that it's not true that all the populations are just sitting there with some constant composition. They're constantly fluctuating. Some bacteria are growing in prevalence, then they're dying, and then another one's coming to take its place. And if you look at it, the population structure is strongly fluctuating, but the metabolic output of the system is not. So you've got the function of this interacting biological system is insensitive to its taxonomic structure. And that's really shocking for biology. It's sort of saying that who's there doesn't really matter to what the thing does, which is really surprising. And what it implies is that the system self-organizes to create a kind of emergent metabolism, which doesn't depend sensitively on the taxonomic or population structure. And why this happens, how this universality in this non-equilibrium living system happens, is still something that we don't really understand. We don't understand it, but we know it's really important because we know it even happens on the scale, not just of a laboratory experiment, but of the world's oceans. So if we're trying to understand the response of the global ocean microbiome to global climate change, we need to understand this universal emergent metabolism of living systems. So is this universality, I've shown you just a couple of examples, is it useful? Well, one answer would be, yes, it is useful. So here's an example of another scaling law in biology. This one was made by bird enthusiasts living in England in the 19th century. So what people did was people in England like to do bird watching. So around about 1859, well, actually, this example here is not bird watching. I'm wrong about that. This one here is plants, but bird watching was also another one. So started looking at, how many plants do you see in a given area? So let's suppose I take a field and I measure how many species of plants do I see. So you go ahead and count them with a botanist, and you find I've got 100 plants, 100 different species of plant. Now I double the area of the field, so I look in the area twice as big. How many more species would I expect to observe? Well, you might say double, why not? But the answer is no, it's not. If the area doubled, the number of species will go up as 2 to the 1/4 power. Not exactly 1/4, but roughly that. And that's, where does that come from? Well, that's an interesting question. I'm not going to go into that, although I do have an answer. This is how it's useful. We use this to measure climate-related extinction risk. You might look on the news today and find that, due to deforestation in the Amazon, 3,000 species went extinct today. Well, I guarantee you that there wasn't a biologist going through there counting all the species that went extinct. What happened was they know how much area was burnt, and so then they can work out how many species may have gone extinct. That's the kind of basis for that kind of estimation. So that's one way to say that universality can be incredibly useful. Even if you don't really understand at the excessive realism level, you can then make extrapolations using these scaling laws. So here's another answer to the question, is it useful? And this answer is no, it's not useful. So let me explain that. This is also an example taken from ecology. So this is an island near Panama called Barro Colorado Island. And it has lovely forests, as you can see. And basically, we know every single tree on this island. Not me personally, but real ecologists. And so you may ask a following question. What fraction of the species, the different species of trees that you can see here, have a given number of individuals? What fraction of them have 100 of those trees? What fraction of them have only 25? Which fraction of them have 400? You know, you can draw a probability distribution for that. And here is the answer. Here are the number of species that have this number of individuals in this, of trees in this island. And you might then say, well, let's see if we can predict those data. So you make one model, and one model is a model that says, well, there's different environmental niches here. Some trees work better in this kind of soil. Some trees work better in that kind of soil, blah, blah, blah. And you can try to make a very detailed model of that. Another way you could answer this question is you could say, I didn't think I know too much about the actual processes going on here. So why do we make the ideal gas model of trees? Why do we make a model that says, well, some tree has to have the most number of species, the most number of individuals. So what kind of, some tree would have to have the next most number. What would we expect that distribution to be if there's just random fluctuations in the populations of these species? And turns out that you can work that out, just like you work out the distribution functions for an ideal gas in statistical mechanics. And when you do those calculations, one curve gives you the black curve, one calculation gives you the green curve. And you can see that they're the same. So here you have a situation where you have two diametrically opposed type of models, one which is based on the process, one which is just based on statistical fluctuations and knows nothing about the process, and they give you the same answer. So we've learned something very interesting about emergence. The other side of universality is that you can make predictions based on a minimal model. The downside is that statistically similar results are obtained from microscopic models that are different in their assumptions. So if you have universality at a high level of description, then that precludes you understanding the lower levels of description. This is the same thing we talked about last time, how it's easy to go from statistical to go from one level of description to a higher level of description, which is what we do in statistical mechanics, because that map is many to one. But to try to understand the basic laws of physics means going from a high level to a low level, and that map is one to many. And we have to figure out which of the many microscopic possibilities are the right laws of physics. So now I want to skip through this. I want to skip through this too. So I'm going to skip through this. I want to go on to this topic here. So I want to end by asking, what do we miss by not having a top level understanding of biology? And I'm going to come back to one of the central topics of this course, which is superconductivity. And I want to try to use this as an example to help us think about living systems. So here is the history of superconductivity, which we're going to go into huge detail about in this class. So superconductivity, the zero resistance of a metal called near absolute zero, was discovered in 1911. Okay, so that's what it means to be a superconductor. 1933, it was discovered by Meissner and Oxenfeld that a superconductor doesn't just have zero resistance, it also expels magnetic fields. And that expulsion of the magnetic field is called the Meissner effect, or Meissner-Ochsenfeld effect. In 1955, it was predicted theoretically and discovered experimentally, that in a magnetic field, certain types of superconductor have flux tubes that organize into a periodic array known as a vortex lattice, or sometimes an Abrikosov vortex lattice. And 10 years later, it was discovered, and again predicted theoretically and discovered experimentally, what are known as the Josephson effects. Now, the question I want to ask is, why did it take so long to make these discoveries? This was 1911, 1933, 1955, 1962, 20 years from here to here, 20 years from here to here, 10 years from here to here. Why did it take so long? When we do this in class, I'm going to be able to derive these things, or you're going to do some of them yourselves in the homework. You can derive them very easily from a few lines of algebra, from the idea that you have off-diagonal long-range order, a kind of quantum order that we're going to talk about very, very soon, of a charged Bose-Einstein condensate coupled to an abelian gauge field, electromagnetism. And all of these things can be really easily understood at a top level of description. But they can't really be understood very easily at a low level of description. So let's talk about the levels of description in superconductors and compare and contrast that with the levels of description in biology. So here on the left is superconductors. So at one level of description, you might be talking about quantum chemistry and material science, solving the Schrödinger equation for the atoms that make up the material. At the next level, is something again we're going to talk about a lot, is the BCS theory for interacting Cooper pairs, electrons that normally repel each other, but in a superconductor, they attract each other through the exchange of virtual phonons, and they form these composite bosons called Cooper pairs, which then interact, Bose-Einstein condense, and form superconductor. And then we'll talk later about Ginsburg-Landau theory and the idea of off-diagonal long-range order coupled to electromagnetism. So those are three different levels of description of a superconductor. Let's think about the levels of description in biology. At the lowest level, you might have atoms and molecules. That's like this level of description for a superconductor. Then there's a kind of functional level description. You know, here we talk about superconductors, the Bardeen-Cooper-Schrieffer theory for interacting Cooper pairs, theory developed here at the University of Illinois. In a biological system now, we're at a much higher level of description. We may be looking at the elasticity theory for DNA, or we may be looking at liquid intracellular complexes inside a cell. That's currently a very hot topic in cell biology. And then at the highest level, the most abstract level, we may be thinking about, say, something like the dynamics of evolving self-programming systems, like we talked about before. So those are three different levels of description. And these different levels of description answer different questions. So in superconductivity, this level of description, the quantum chemistry and material science level of description, is good for answering how do specific materials instantiate the BCS mechanism of superconductivity. The BCS theory for interacting Cooper pairs answers the question, what is the basic mechanism in weakly coupled superconductors for superconductivity? It doesn't answer what happens in high temperature superconductors. And the Ginsburg-Landau theory, the off-diagonal long-range order coupled to a gauge field, explains for you why does the phenomenon of superconductivity exist in the first place, and what are all the consequences of the existence of that phenomenon. And that's understood by the high-level description. So if you were to ask about, well, what could you answer in biology if you understood these levels of description, you might make the list as follows. The atoms and molecules, that can tell you how do specific biopolymers interact? How do they fold? How do you get template-directed synthesis to make the process of translation, the process of transcription, these very molecular cell processes? The elasticity theory, the understanding of intracellular complexes, can tell you something about the basic functional cellular process at a large scale. And the dynamics of evolving systems can tell you something about why the phenomenon of life exists at all and why you get this open-ended growth of complexity. So basically what I've suggested is that the different levels of description correspond to different levels of universality, which are good for answering different questions, summarized here on the left for superconductivity and summarized here on the right for biology. This level of description is good for describing specific materials. This level of description is good for describing weak coupling superconductors, but definitely not high temperature superconductors. And this level of description of symmetry and gauge theory description is good for describing all superconductors. Similarly over here, if you understand about the atoms and molecules in a living system, it'll help you understand what happens for specific biopolymers, such as a tRNA or something like this, or a protein. If you understand this level of description, it will tell you something about the physics of the subcellular components. If you understand this level of description, it will tell you about all of life, the principles of life, which transcend the instantiations that we have on Earth. So why do we need this universal level of description, this large-scale level of description? Well, in a superconductor, that high level of description, which applies to all superconductors, what it does is it tells you that you would be able to predict the response of a superconductor to electromagnetic fields. In one fell swoop, you understand superconductivity, the Meissner effect, the vortex lattice, and all the Josephson effects. All the phenomena that are associated with superconductivity, you can understand them at this high level of description. And if you don't have that high level of description, you can't understand or even predict or even discover these effects existing. So the fact that as in physics, we started off at this sort of most obvious level of description of sort of atoms and electrons and things like this, and not really thinking at the high level of description, we missed the most important features of superconductivity. So in this course, why we're trying to teach this higher level is because we really want to understand the universal emergent properties of matter, not specific instantiations. Now, when we talk about biology, we think that this high level of description would apply to all life, dynamics of evolving self-programming systems, let us say. What happens if you don't have that? Well, in a superconductor, if you didn't have this high level of description, you couldn't predict the response to electromagnetic fields. All of these things, the Meissner effect, the vortex lattice, the Josephson effect, the superconductivity itself, all of these things are what happens when you hit a superconductor with an electromagnetic field. So we want to understand the response function of a superconductor. What happens if you try to perturb not a superconductor, but a living system? Well, the way we perturb them is we try to make a selective perturbation. For example, you've got a bacterial infection, so you give somebody an antibiotic. You've got cancer, so you give somebody chemotherapy. You have some weeds growing in your field, so you apply herbicides to kill the weeds. Or you have too many bugs coming and eating the crops, so you apply an insecticide. So the question is then, how do living systems respond to these types of perturbations? Well, in a superconductor, what happens is by regarding superconductors reductionistically as collections of atoms, what we missed were the emergent laws that act at the system scale and govern the large-scale response to electromagnetic fields. We know how to solve that problem. The analog problem in biology is what happens when you apply these things. And the answer, as you surely know or suspect, is that the biological system evolves to defeat your perturbation. You try to apply antibiotics to living systems, and very quickly those antibiotics stop working. Within not millions of years, the usual timescale associated to evolution, but within 5 or 10 years, maybe less, globally. Okay? If you would try to apply chemotherapy agents to somebody who has cancer, those cancer tumors evolve rapidly and respond to the chemotherapy agent, and you get chemotherapy resistance. If you apply herbicides to try to get rid of the weeds, eventually those weeds become stronger. You kill the ones that are not able to survive, and you end up evolving stronger weeds, and so on. So whenever you have a biological system and you intervene in it by treating it like a physical system, it backfires. It doesn't work. The biological systems reprogram themselves as a system and evolve around the perturbation. So what happens is that by regarding biology as complicated physical systems, we're missing the emergent laws that act at the system scale and govern the large-scale response to control perturbations, except that those perturbations aren't electromagnetic fields, they're attempts to try to limit bacteria or cancer tumors or whatever. So we know how to solve this problem because we have a top-level theory. We understand how to write down and describe a superconductor from the point of view of off-diagonal long-range order, symmetry, and gauge fields. We have no idea how to do this for biology, and we need to know it if we're going to understand to control biological systems. So what I've kind of suggested is this. There's a kind of obvious hierarchy of sciences. Here's the physical sciences. As Dirac said, chemistry is just a subset of physics, and biology is some mixture of chemistry and maybe some physics as well. That would be how most people would draw the hierarchy of the sciences. I would draw it the other way around. I'd write it like this: biology at the top level, then physics and chemistry. Why? Well, because my point is that in biological systems, you're looking at systems that are able to literally change their Hamiltonian, which is what you mean when you say they reprogram themselves. They actually rewrite the rules that govern their dynamics. Physical systems are the set of systems that don't do that. So they're a subset of those other broader class of systems. Okay, so in physical systems, you have predetermined dynamical rules, e.g. from knowing the Hamiltonian. In biological systems, the rules themselves are macroscopic rules that operate at the level of the biological object. Those rules are themselves emergent. So that's a higher level of emergence beyond anything that we're used to in this course, where the actual rules, the actual laws of nature are emergent, and that happens in biological systems. So dynamical rules can be emergent. That's what makes biology hard to understand, biological systems hard to control. And so in my view, that's what makes biology an interesting physics problem. It is right at the frontier of physics. We're looking at systems that have a property that the usual physical systems don't have. They have emergent dynamical rules. And I think it's a super fantastically interesting problem to try to understand that. So I'll end my introductory lectures with this quote from Stanislav Ulam, who was a mathematician and worked at Los Alamos National Lab for much of his life. He made the following quip. He said, and he was parodying John F. Kennedy, who in a famous speech said, "Ask not what your country can do for you, ask what you can do for your country." That was an inspiring speech given by President John F. Kennedy. Ulam says, "Ask not what physics can do for biology, ask what biology can do for physics." Meaning, usually we sort of think that physicists can help biology. We can make better microscopes, better computer simulations, better computational and statistical techniques, and so on and so forth. But really, the interesting thing is that the biological system actually pose a truly interesting and novel question for physics. And so biology poses a way to actually expand the scope of physics. And so that, I think, is really the most interesting example of emergence. Okay, so it's the time to end the lecture, and I've come to the end, so we'll leave it there, and I'll be happy to take questions. So what I've told you is in this introductions, emergence arises in large systems due to strong interactions between constituents. A collective state exhibits novel response characteristics and loses memory of its underlying level of description. The onset of emergence is accompanied by phase transitions and scaling. Emergence is accompanied by generalized rigidity. The emergence of patterns and topological defects have interesting scaling laws, and one can try to understand these in the same way that one does for equilibrium systems. Minimal models, emergence can be exploited for the purpose of quantitatively modeling very complex systems. Emergence yields important universal predictions for how systems respond to perturbations, because one can understand a top-level description, which in many ways is simpler than the more microscopic ones. And biology is a wonderful frontier for studies of emergence in physics. Okay, so that's the end of the lecture, and I will be happy to take any questions.
By the way, I mean, when would we have our first homework, and where can we find it? I mean, I checked the website, but I only found a homework for a earlier course. But I mean, yeah.
Okay, so that's so. I'll answer that question. I'm going to just pause the recording.